So
Okay.
a tiny bit about the background of what we're doing. So we're studying transgender youth. Transgender youth are youth who don't identify with the gender that is incongruent with their sex assigned at birth. And we know that there are certain gender and sex differences in spatial and social cognition that can be observed both at the behavioral and neural level, and we want to see if there's any differences in that regard when your gender identity doesn't match your sex assigned at birth. Oh. Sorry. Oh, okay.
Oh, okay, how it works. So our research question is whether the brain activity of transgender youth who have not gone through the gender affirming treatment, their brain activity during the performance of tasks such as mentalization and facial emotion processing, whether it's showing the gender differences more congruent with their sex assigned at birth, or whether it's aligned more with their experienced gender identity. So we have 4 groups of interest that we're going to compare cross-sectionally. So we have birth assigned male transgoes, which we say trans AMAP. And we also have the trans AFAB, and for the cisgender controls, we also have cisgender AMAP and cisgender AFAB. So those are the 4 groups of interest.
Yeah, AMAP and AFAB is just like
at birth.
Okay. Okay.
Okay, so the data that we have collected are functional images for the 2 tasks, mental rotation and passive viewing of videos showing people expressing different emotions. And obviously we have structural images to register those functional images too. And these tasks were selected particularly because they have previously shown consistent behavioral differences depending on sex, especially mental rotation. Task is like the only one that really survived all the meta-analysis. And there's also some studies that show differences on the brain level. And we collect the data at
so the participants would go into the scanner, and on the left side we have example of the mental rotation activity. So for this image, participants would view 2 block figures, and they need to mentally rotate these block figures in their head to see whether they're the same but rotated or mirrored image of each other but rotated, and they indicate which one it is using a button box. And on the bottom here is kind of a control condition where they just indicate where the arrow is pointing. And when they see a triangle, they just don't do anything. So on the right side, it's I'm gonna play it. It's the example of the viewing of the faces. So participants would view faces that start with a neutral emotion and then turn into a negative emotion. And or another condition is they would from a neutral condition and turn to a neutral condition. And as a baseline activity kind of condition, we just have like moving circles.
It's kind of like
Yeah.
what it's gonna do to our
Yeah.
participants.
It is weird. I worked with that one. Never mind. I'll come back to it.
Okay.
Yeah.
Okay, so we want to learn as we process these images. So we're gonna use processing pipelines and tools that we haven't used before. So for the pre-processing, we're aiming to use fMRI prep. For first level modeling, we want to use NiLearn, and for group comparisons, we actually don't know yet what we're gonna do, but it's part of the learning process, I guess. And we want to perform all the analysis using Scinet, the supercomputer.
So for our objective of our project, we want to use part of our data that we have collected to conduct preliminary group comparisons of brain activities during the mental rotation and also the phase task. And for the deliverables, we hope to put together some documentation that has like the processing and analysis workflow, some steps of the quality control, and also some links to resources so that we can train ourselves and hopefully train other members of the lab in the future.
Okay, and yeah, we obviously need to thank our beautiful team that are actually more responsible for that project than we are, because we joined after the project was already like designed. And if you're interested, here are some references to papers about sex and gender differences and cognition and gender dysphoria and neurocognition.
Thank you. Any questions?
Are you collapsing the trans and the cis groups just into two groups? Are you doing it, are you stratifying it by, for example, sex at birth being boys versus girls? So is there 4 groups or is there 2
Four
groups?
groups.
Is your sample size much smaller?
Right,
Yeah,
I think the aim is 35 people per group, but the trans AMAP group currently only has like 20 something. And there, some of the groups haven't reached 35 yet, but we're still trying to reach out to people and collect more
So
data.
this is in progress.
Yeah.
So that's also why we're analyzing, here we're gonna analyze our baseline data, which makes it a cross-sectional and also pre-treatment, but the final goal is to do longitudinal analysis and at 3 time points, so before treatment, after puberty blocker treatment, and after gender affirming hormone treatment, so either testosterone or estrogens. Gender.
Yeah, but there's so many more like treatment trajectories now, so we're trying to explore those as well.
I was just curious, like, for example, if you collapse just for the, like, for power purposes, if you collapse all the time and all this stuff. And then if you found a significant difference there, then you stratify
Okay,
by either,
but we can't really combine them, because then we would have, if we combine the trans, then we have trans assigned male at birth and assigned female at birth, and if we're if like.
You're looking
like
for the
our-
sex differences in
Yeah,
cognition.
we assume that there's sex differences in cognition, then they're different people. So in terms of like their brain,
Yeah.
right? And the same like with cis boys and cis girls, like we assume that they're gonna be different. And if we were to collapse groups, we would probably maybe also do analysis where we just include AMAP and AFAP people, independent on their gender identity. But that kind of doesn't really answer our research question, so it would be more maybe like to explore if we don't see any differences.
yeah, like.
Yeah.
Are there any field maps or reverse phase encoded images?
So unfortunately, well, for me, unfortunately, I don't know, maybe it's great, but the field maps were collected in a way that they're like the forward reverse images.
Yeah,
So
yeah, so you should incorporate those.
yeah, so
Yeah.
we want to definitely,
Okay.
Yeah.
Because
because there's 2 ways of collecting field
Mhm.
maps, right? And it's like
yeah.
the one that only doesn't work with the SPM pipeline, that's why. I'm
Oh,
happy.
no, but it's more robust with that more prep
Yeah?
if you get them
Okay.
coded properly. Yeah,
That's good.
because the original like mag phase field
Yeah.
maps, I've never gotten the settings perfect to get them to work.
Okay.
But
because
the
the
reverse
like
phase
the
encoded are pretty robust. Yeah,
So that's a good,
yeah,
good thing to know.
Yeah.
Better.
Yeah, yeah, and usually it's a trick to just make sure that they're named properly in the bid structure so that
Okay.
incorporate them properly.
Okay, great.
Don't you want to say something about the video about the samples?
yeah. So no, so I actually that's totally the task that I worked with like in my postdoc. But I remember actually the so-called neutral phases
Yeah.
are actually more interesting to look at than the angry ones because
of activity.
yeah, or in terms of. I mean, actual as a participant
Yeah.
too, because they like, because they do a different facial expression, in the so-called neutral, they do a different. Yeah, they do, but they called it neutral, so they just asked them to do anything with their face. So they're like sticking out their tongue or puffing their cheeks or doing something odd, and every actor decided to do something differently.
So I guess that's
So-
not very good control.
So it's not, that's what I was gonna say.
But
Because it actually like, you start to see habituation to the angry, because your brain is like, oh, this person, oh, all the people are gonna be angry in this block. But for the ambiguous, the brain still doesn't know what the person's gonna do, and you actually get more sustained activity in a lot of the facial processes, so it'll be interesting to look at what you're like pointing out.
And those circles, my interpretation is that circle is just,
Oh, they're very hypnotizing.
yeah, I know. I thought they're just there so that you get kind of a baseline on how your brain processes anything that's moving on the screen.
yeah.
It's pretty much, yeah,
Yeah.
it'll be interesting to see what you can pull.
Yeah, I mean, to be honest, like, personally, I'm worried about the task because a lot of our participants. A lot of people actually fall asleep.
Yeah.
That's one problem. And the other problem is that, I think that generally social processing tasks don't really show, gender or sex differences that are that robust. So even if the task was perfect, there is a high chance that you wouldn't see anything with, especially like with the
Yeah.
size of the groups that we have. So I'm generally more hopeful about MRT because it's a better task and
Yeah.
the people actually need to do something. So we kind of
Yeah.
like know whether they were asleep or not.
Yeah.
It's not just the notes
Yeah.
from the MRI,
Mhm.
but it's like, you can see if they press the button.
Yeah.
And they fall asleep during the circles?
They fall asleep during
All the
the
time.
Everything. Yeah.
Even to
Yeah.
MRT.
It's just like.
Yeah.
Fall asleep.
Oh,
Yeah, but yeah.
MRI, nap time, great.
Yeah. Well, at least MRT, they were supposed to click a button like every
Yes.
second,
But
so
not
you
with
know
this
when they fell
button.
asleep, because there's no button presses in.
Okay.
Yeah.
But imagine you're in the scanner, it's dark and
It's
cold
dark
and
and
feels
it's
like
cold.
You fall asleep every time.
I fall asleep every time.
You have fall asleep earbuds.
Yeah, I've napped in there personally, but.
You fall asleep during your task.
Yeah,
Yeah,
I think our, the design, the order in which we're, I don't wanna like poo-poo on my own study, but I didn't design it, so I think the order in which we do the scans might not be the best, and we cannot change it anymore. The rationale was to do the resting state fMRI first, because that's the most difficult to keep concentrated.
Mm.
And we
To get it out of the way and then
Yeah.
do the
do
task.
2 sessions of resting states, not just
Oh,
one, 2
yeah.
sessions, 15 minutes.
And because like it has been designed this way, it lulls people to be very tired and hazy after the resting state, and when they get to task-based, it's kind of like a hit or miss. They'll either just like
It-
fall asleep.
so for someone that always falls asleep in the scanner, like me, I can tell you, I start falling asleep during the anatomical scan, it doesn't matter.
Okay.
I'm never awake.
Okay.
The anatomical.
Later.
Yeah.
Yeah, because
Yeah, like the moment I lie in the scanner,
it was.
1 minute in,
Yeah.
I fall asleep.
Okay.
Okay.
Maybe it took me on a plane. I
Yeah,
fall asleep
so-
before takeoff.
Oh, okay.
Exactly, okay.
Yes, in the interest of getting through everyone
Yes.
before 5. Who's
We have
next?
Catherine?
For fun.
Sure, I just have to.
Yeah.
Open the
Yeah.
Zoom link. So.
Oops. Oh, okay.
Actually, no, I have to.
Mm.
And I have. to
I'm still using my old bathroom.
Oh,
I'm all set, I'm just joining now, so then just make me co-host.
so,
Is that how-
Everyone should be
I
able to
think you
share
should be
now.
able to
Oh,
share.
okay.
So, Samantha and Efra are going next, so be prepared.
Okay.
They're able to share.
Yep, there we go. Should I keep mine muted, because the owls?
Yeah, keep yours. muted.
Okay, there we go. Hi, everybody, I'm Catherine. I work in the Cognition and Aging Neuroscience Lab at York University with Dale Stevens. Our lab really, a lot of us look at resting state in some way, shape, or form. That's kind of what we all do. So for my dissertation, I've been looking at resting state parcellation and targeting methods for TMS, but I'm really also interested in this thing that keeps coming up, which is brain-derived neurotrophic factor genotypes, and how the BDNF genotype has kind of become like this like boogeyman in all areas of research. So I'm really interested in looking at if I can look at the resting state functional matrices that I have and compare the 3 different genotype groups. So that's what I'll do a little overview of today. Okay, so the Val66 MET is a single nucleotide polymorphism, or SNP, of the BDNF gene, and this has been linked to like atypical hippocampal size is probably the biggest thing, which is then relates to memory. So there are replicated effects of this gene, but because it's had it's robust in some areas, it's starting to be kind of implicated in a whole host of different pathologies. So up in the corner, this is what it looks like, as if we would know. So basically what you have is 2 alleles, and usually you would have a valine allele, so Val homozygous participants, like right here. This is what we usually see. This is like the most prevalent in the population. And then what happens is that codon 66, we get this valine to methionine so we're looking for translation, mutation, that's the
Translocation?
Yeah.
No.
Mutation. Polymorphism.
Polymorphism, yeah.
I'm not a geneticist. So sometimes when we see this polymorphism, we see like a heterozygous mix of alleles. But we also do see where people get 2 copies of the methionine allele. This is incredibly rare. In Caucasian samples, it's about 1 to 3%. But this methionine or having a being a VALMET carrier is the majority in a lot of Asian populations, especially in Korea. I think it's about 23% of people have 2 MET alleles. So I'm kind of interested, a lot of studies will also collapse these 2 groups because there are so few MET carriers. It's hard to know if there's a compounding effect of this allele as well. So I'm kind of interested in using a big sample to see if I could have enough people to actually look at this. So this is from a review paper that has looked at everything BDNF has been implicated in. So specifically, I'm interested in this is like intrinsic functional architecture of our networks differ across these genotype groups. So what I've processed so far is the Human Connectome Project data. So I'd like to use this again. I already have the matrices generated. I use the Schaefer 200 atlas with the YOS 17 network parcellation. So all of my participants now have individualized parcels that are also like comparable across them. Yeah, so I'm really interested in this data set because it's so large. It has multimodal data. And things like repeated measures. So the resting state, there's 4 different scans collected on 2 days. And so the resting state data, I've already pre-processed it and parcellated it. So I'm looking for my tools and methods. I'm so inspired by Richie's talk yesterday. I'm like gonna jump into PLS, but the regression version. And then look at like support vector machine learning classification. So I'm looking at taking my matrices, I won't go through all this, taking my matrices, running through PLS regression to kind of get these components of importance, and then looking at using a classifier. Because there's a repeated measure, I have training and test data, and there's a large sample as well. So using a classifier to see if it's able to pick out any genomic differences. So these are just some details of a little bit of what we learned yesterday, and how like just different cross-validation techniques that I'm interested in. And I'd like to ideally host all of this, I'll get to that, it's a slide. I remember putting the image in now. Yeah, so my objective is to leverage this large data set to investigate the possibility of distinct functional profiles. And this is really important to me because this boogeyman idea is also a very racialized idea. And I don't think we have one prototypical resting state architecture. We probably have many different profiles, or I don't know what to call them, but kind of different patterns. And not one might be more pathological than the other. So I think this question is interesting because it looks at if there's a genetic influence on resting state network architecture and to what extent, because this is looking at one single SNP. So one single SNP could change our cognitive architecture. That's important to know. Or this other idea of can this show if resting state network configuration is more experience driven? And this is also why we see changes in resting state across the lifespan. So in terms of deliverables, fingers crossed, I would really like to have this in a GitHub repository with a Docker container, so it can be used by whoever would like, and have a Jupyter notebook. Especially our lab is really focused on mentorship, and right now we're all late PhDs, and so we're kind of worried about knowledge translation as we start to leave. So I'd really like to have something like a standard operating procedure and just get this kind of process of getting things on GitHub more adopted by my lab. Some references, these are mainly all the BDNF ones. And that's it.
What do you plan to, so if you're doing PLS regression, so you're trying to predict, I guess, your resting state connectivity with the three groups of alleles, is that what you're?
Yeah, I think that like I'll be looking at like the genotype group, like their alleles as one kind of data set, and then looking at the matrices that I have. And I'm curious if like with the regression, if it'll kind of pick out the central parts instead of like everything being a whole brain analysis. But this is from this morning, so I have a little bit more digging to do.
So,
Could try to the status.
oh, yeah, so there's another thing we can do is you can extract dimensions also from the connectivity, and then narrows down to like the characteristic pattern for systems, network systems. And then you could focus on that. That's one thing. One other thing is you could consider just starting with PLS correlation, because PLS regression are a little bit more complicated. And it's not as straightforward as PLSC, and also the first dimension is basically the same.
Oh, okay.
The other thing is if you only have so for the genes, I would expect only 3 columns, right? Because you have 3 types of SNPs.
Oh, okay.
So
Yeah.
that
so
only
okay.
gives you 3 dimensions max. So the number of dimensions you could get will be the minimum number of variables between the 2 sets. So if you have something that has more dimensions, then you might miss that. But if you have a very strong effect, which I'm guessing you will have a very strong age effect in the first dimension, and
Yeah.
that might be dominating your analysis, then
Probably, yeah.
that is
probably
okay.
need to check on the initial.
yeah. But I would, one way to, you could run it first and see how the dimension looks like. If you have a very dominant dimension or 2 dominant dimension, then you're good. But if the 3 dimension looks too similar, with, in terms of their explained variance, then you might want to increase dimensions to be able to get all the signals you want, and to do that, you can consider adding some control SNPs just to increase the dimensions.
Okay.
That's great.
I was hoping to add more SNPs, but right now, with the NIH, our genetic goals are, we're waiting for ethics review, so I can't access any more data right now.
No, you can start with
Yeah.
one,
Yeah,
yeah.
Okay.
well, and that's cool to start with the SVM too, but what do you have for what proportions do you have in HCP for these alleles?
I think it's about 45%. And this
Oh,
is,
okay.
this is including related
okay, so if
to
you-
the full sample is about 45% Val,
Mm,
about, I think it's like 4% Met, and then
Okay.
the rest are heterozygous.
okay.
So it's,
No,
it's a
that's
small,
what
we've tried to use if we've looked at unrelated BDNF participants,
Yeah.
we've done propensity score matching and all these comparisons, and we found nothing. So it's kind of, I mean, I
Yeah.
really want to be like, okay, let's like look at a big, like all
Yeah.
pictures, or something to see if
Yeah.
there is anything, because this network effect's only been found in children and never replicated.
Oh.
So.
Okay. That's cool.
Any other questions? No? Okay. Very cool. Let's get to Sam and Ifra and Jexter and Clara this next. The volume on?
Let me myself.
Yeah.
So.
Yeah.
So for reference, we switched our idea like 3 or 4 times, and we came up with this one, I guess, 20 minutes ago or less.
Still working on it.
Yeah.
but-
Still thinking about, what we're doing.
Yeah.
So, we're using the HCP data set, so just like the general one to make the project grow with like 1,000 participants, rather than, we're gonna do psychosis, but we decided to put on the larger majority. Initially, we were supposed to start with, I think we wanted to look at sex markers. But it's still in the loop, I guess, we could go back to that, but now we're trying to focus on, machine learning regarding cognition, so the behavioral data sets of like each participant and their scores and comparing your machine learning to them. That.
And we can get your guidance on that. Still trying to iron out the kinks. Some background on functional connectivity. What we know about it, machine learning, my 5 minutes ago. Right up on it, but we do need to do a deeper dive on it. Yeah. Thanks. Okay. Some of our main questions, if you do see a whole brain analysis there, just disregard that. We were, making changes. So overall, like, how does the brain connectivity region, so the regions that are yet to determine, but maybe possible hippocampus, amygdala, like subcortical regions. How do they change over time? The functional roles of different brain networks, more specifically, machine learning techniques be applied to predict like individual differences in functional connectivity. Again, within the regions that we will probably get to. Okay.
So as Bruce said, we are using from data, it's between ages of 18 and 30. We actually were thinking of still using the psychosis set if it is open. It is a shorter age range, 20 to 25, but I think that data would be really good to use as well.
Maybe we can do something with that.
We'll see how that goes, but I guess it would be interesting because the psychosis set would be obviously the result of the cognition would most like would there be a difference as well? It's a good question to ask.
Yeah.
here are just, I guess, some tools and methods. that we've been
Not
using.
all of them, some.
Some of them, so all the HCP data has been pre-processed, but we might, depending on if you guys would prefer pre-process it. Go through 50, but, yeah.
It's already in 60.
It's already set, okay, then roll with that.
Yeah.
We'll work with that. And so, yeah, we're trying to get, I guess, a better understanding of static connectivity, in general, and trying to explore those different patterns, with. Please ignore sex markers, it's now changed to cognition, but we might go back to it again, we'll see. Looking at the regions of interest, overall connectivity. And I believe that's the last slide. Oh, the logos. Yeah, so I really want us to visualize the data, so I want to make a correlation matrix. We could make it specifically to like the regions of interest, which would be like, you guys can see like this region, like the default region here, we focus on that. I don't know, I don't think we'll focus on the default region because it's cognition, but it's nice to see, it's nice to look at, it's pretty design. This is modularity here, and so see how they interact together. I guess too broad of a question to ask since we're focusing on cognition, but again, there's things to do. Yeah, that's about it. Great. Any questions?
Of course, reaching is large, but
No, but I do have one suggestion. So with regard to your main questions, I would narrow it down to one.
okay.
So, because 4 is a bit too much to do in 2 weeks.
Yeah,
That is like
Pick
a
one.
year project.
yes, all of those are like general theories of 20 years of science.
Yeah.
So you need to pick a smaller goal.
Okay.
Yes.
So one research question and maybe just one objective. That might be easier to.
Yeah.
To go through.
Cause you're gonna notice also every step towards one question is gonna be a new learning opportunity.
There's any more questions?
And it's gonna bring you more questions. Yeah.
Any other questions?
The crowd. What do you think about the different, data sets like the psychosis and?
I'm still trying to, okay. I still feel like psychosis is only on the NDA. I think Colin is Yeah. confused. And the adults you can grab on Brain Life right away. So I would definitely start with the adult. And then we can look more into whether the psychosis is technically an NDA application. Because an NDA application, so NDA is a NIH data archive. And they put the data usually under the controlled access, so you need to make an application, and someone needs to approve it, and by the time they approve it, the school will open a for you. If we're technically can get to the data because it's in our lab, but then it makes it awkward for both of you guys to work together. So I'd start with the HCP data, because I think that one technically has controlled access, but like, it's like you get the account and then you can download the same day. And or you can just go direct on Brain Life and run it. Yeah.
And for learning process, that's.
Yes, plus there's a lot of it, and if you're interested in relationships to cognition, the cognitive barriers I think are relatively easy to get to as well. Yeah,
So the psychosis data also have a lot of.
you don't know yet, yeah, actually, that's it for the summer,
Yeah.
is figuring that out, but they all have motion, but yes. People who volunteer between the ages of 18 and 35, getting their brain scans for 2 days straight, are usually the ones who are willing to move the least. Versus people recruited from actual clinics struggle a little bit more with it.
So you might want to deal with that in real life, but not in the 2 weeks of brain hack school.
Yeah.
Okay.
Mm-hmm.
Any other questions, thoughts?
Cool.
Okay. We'll go with Ernest and Lucy's group. And then the rest of it.
Should I take my system mute?
Yes, mute.
Hello everyone. The title of our project is characterizing resting state, EEG and state of preparation. I'm Ernest. I'm from SickKids, also a teaching candidate at UFG.
Hi everyone, I'm Lucy, that's the Robin Research Institute this summer. So to begin, state information is very prevalent in modern society. I'm sure many of us in this room have experienced that. There's significant implications with state information for the physical, mental, and cognitive health for the individual. Investments in community that it captures neuronal activity within the human organism. And so previous studies have demonstrated that deprivation has been linked to alter certain data and activity, which are linked to deficits in executive function. So it'd be interesting to look at the differences between the two groups and see the information or not. So in terms of research objective, our general research topic is characterizing patterns in resting state EEG data in sleep-deprived and non-sleep-deprived states using unsupervised machine learning. There are three sub-objectives. First one is statistical analysis and data exploration to compare the sleep deprivation and sleep states. Second objective is to identify the EEG patterns associated with sleep deprivation. And the third objective is to compare the effectiveness of different unsupervised clustering techniques. In terms of the data, we found a database available on the internet, and within this database, there are 71 subjects that were included. The age range is 17 to 35 years old. There's a very strict inclusion/exclusion criteria listed in the slide here. And the study is conducted using a living subject design. So we begin with the subjects in normal state, and then we have the EEG data measured. And then another 24 hours sleep deprivation session, and then the EEG study.
So we're gonna use different tools. One thing in terms of tools and methods and the objectives is that we're not gonna promise anything. Learn through years of study in medicine is that we cannot guarantee it will work, especially for this data set. We have a very limited sample size, so we'll see what will happen. However, for now, the plan is first to repeat what was provided in the reference paper. So for that, we will use violent plots, and we have data visualization for sure. Map is the starting point, and then we will go for unsupervised ML methods. In terms of deliverables, documentation, and the whole structure of GitHub repo. The paper is very recent, so we hope it's gonna be fine. Another thing is that we actually started the analysis. So, in terms of the violent plots, we could actually repeat what was provided in the paper, which is good. However, for UMAP embeddings, we did not observe any clustering or separation. We will try to fix this through more in-depth ML methods, feature engineering, data augmentation, different things. That's where we are.
Any questions, comments, suggestions?
I actually have a question, if that's okay.
Yeah.
Thank you. Great presentation. So I just have a question about the use of supervised machine learning techniques. I was wondering why you're not going with unsupervised ones. Is that because you want to cluster them into like K clusters, or is there a specific reason you're going with supervised machine learning?
Thank you for the great question. Do you hear us clearly?
I'm
You just asked a question, you don't think?
Can you review it?
So, a great question. I hope, my voice is clear. In terms of, supervised methods, we will use it. Well, between you and me, we have used it.
Mhm.
The only reason we didn't touch it, in the, project pitch was that the data set is very small.
Mhm.
So we were not sure if it would work. And so far, it has not worked.
Hmm.
So with just 71 subjects, it's a tricky project.
Mm.
We'll see, but it's a great idea. We have, some demographics information from the subjects. We also have EGs, so we will explore that avenue.
Mm. So it's interesting too, because it's like but it's a within subject design. So sometimes in these like EG within subject designs, I always wonder if it's like almost the N of one version of the supervised learning actually works better than the full group version, because if you're looking for the intricacies of the differences within one person, versus just trying to find like a signature of sleep deprivation across the group and ignoring the actual fact that you have repeated measures.
That's a great point.
Yeah, yeah. That's the thing. I'm not even, can't even think about if there's like a good supervised method that separates within connected classes.
Not sure.
So this subject, you like measure the people's use the EG to measure people's, I guess, when they're like slept or when they haven't slept. It's after one night of sleeping, and then so that's the sleeping condition, and then the next night is no sleep. And then, oh.
Mhm.
The whole night.
Yeah, and then they hate you, but they still do the EEG somehow. And they.
It's all the whole night. But I'll lie down.
Do you like have camera? It's very fun.
I guess so.
Awesome. You do?
Oh, no, this is, we got the data from the, percent.
Okay.
They're called.
I mean, they're paid.
They're paid.
It's nothing fun.
Like, how much, like, is it?
Yeah.
Not eight,
It is funny though, when you get these studies, like. Some of them, they keep paying, but they give you an out, and it's like amazing. There's a certain point where like nobody is willing to stay in a sleep deprivation state, like if they let it keep going. Cause it actually like starts inducing like terrible mental health and psychosis at some point.
Yeah.
And becomes a form of torture.
Yeah.
Yeah.
But if you're not in the study and you're just at home, it's easier, I guess.
Could like recruit maybe like, residents on call because they're sleep deprived.
Yeah.
And that like, they don't have to be. paid.
Yeah. Maybe that's a really just want to put money.
But I think it's different because for residents or people that are working, they're
Mhm.
occupied by something instead of just trying
No,
not to
they're
fall
not.
asleep.
They're not always working. Sometimes they just have to stay up.
Yeah, no,
in case
it's
they
true,
don't
actually, I think I remember, I knew a friend of a friend, our friend was describing that she had a colleague who did sleep deprivation, or she was collaborating with someone who did sleep deprivation. He said they put them in a hotel, and most of the participants ended up being med students who needed to study anyway. Who were just willing to like sit in the hotel room and study and
Be
work
paid.
and be paid to not sleep.
Sometimes they don't have to do any work, they just have to stay up. in
Yeah.
case they get a call. I think very long ago, someone did a study
Yeah.
comparing their brain and the brain of like someone who is like disoriented and they
Yeah.
found similarities. Which is after which they said that
Mhm.
you can't have false as long as they use.
Yeah, they completely changed on call, because they realized it's like screwing up people's circadian rhythm, so.
It was like compromising patient care.
Yeah. Okay. I got it.
Next one. Jetsera Terrace.
Yeah.
Yasra is next.
Hello?
Okay, I'm just gonna share my screen.
Oh, wait, it's not like. Yeah. Oh, it's fine. Although, are there people who need to go because they need to leave? Okay, okay.
So can you guys see the screen?
Yeah.
All right, so my name is Just working on predicting dementia progression using machine learning on longitudinal MRI data. So dementia is a group of diseases that results in cognitive decline, and generally the pathophysiology is characterized by aggregation of misfolded proteins. So, for example, we see amyloid beta plaques and neurofibrillary tangles in Alzheimer's. More than 55 million people worldwide have dementia, and it's more common in the elderly. And early detection is important because it allows for timely treatment, which can improve outcomes and quality of life for the affected individuals. So the main question is, can we develop a machine learning model that predicts the progression of dementia using longitudinal MRI data? And this is the question I'll be starting with, but I'm aiming to hopefully expand this after looking at the data set in more depth. So the data set I'll be using is Open Access Series of Imaging Studies, or OASIS, and they have a specific set of longitudinal MRI data in non-demented and demented older adults. And in this, there are 150 subjects with visits separated by at least 1 year. And the majority of subjects saw no change throughout the study. So in other words, they started and ended either non-demented or demented. And there was a small group that started non-demented and developed dementia by the end of the study. So the language I plan to use is Python. And some of the libraries I have in mind are scikit-learn, matplotlib, and nylearn. And so I'll start with the data, pre-process, extract features, and finally train and validate a machine learning model. And the objectives are to develop a machine learning model to predict dementia onset, identify early biomarkers from T1 weighted MRI scans, and enhance our understanding of neurodegenerative patterns in dementia. And by the end, I hope to have a GitHub repository, a brief report, or a presentation. And these are the references.
So I forget, are you managing, how are you pulling in the OASIS data? Is there a nylearn fetcher for it? Or you
Sorry.
already have? So have you already downloaded the OASIS data?
Yeah, I made a request and just got it.
Okay. And do you have an idea of what you want to do for the pre-processing?
I still need to look into it in more depth. But yeah, I'm not sure yet.
Okay. And then your machine learning, you're planning to just do it on the full images? Or on features?
On the features.
So do you know which features?
No, I still have to look into the data set. Like I just got it. But yeah, I'll see.
Cool. You guys want to go next? Yes. Okay.
I think I can start with asking us well as a bunch of the slides. So we're all from the same lab and we're all MSc candidates at the Institute of Medical Science at UFC. We were initially planning to use our My Studies fMRI data with within subject differences between insulin versus placebo patients in individuals with depression and bipolar disorder who are performing a reward motivation task, where we can see their effort expenditure for rewards essentially. Unfortunately, due to UH and privacy concerns, uploading the data onto GitHub and everything, we're not able to use that data. So we're going to be still doing something related to our field, but using open source data.
Okay.
But before we get into that, I just want Hattie introduction as well to use their own thesis projects, which are based on EEG.
Okay, so mine, I'm comparing EEG spectral signatures of psilocybin and ketamine and treatment-resistant depressive suspense. Yeah, that's fine. What about you?
mine is going to be a crossover study looking at differences between MDD and health controls, specifically looking to see if there's any correlation between EEG correlates, specifically spectral signatures, and just power band analysis with metabolism biomarkers. So the max, just carbohydrate release, things of that nature, using a metabolic card.
Okay.
And then just doing that crossover between if they're taking a glucose drink, so essentially just a sugary drink, versus a placebo drink. So seeing how glucose affects that in terms of the willingness to expand effort on at the base level, similar to Anika's with looking at anhedonia and motivation and things of that nature. But that has yet to be started and collecting that as well.
Yeah, so for this project, we'll be doing something very simple, still in our field, but we'll just be using open source data to look at whether EEG spectral signature differs between high severity versus low severity depression in college students.
Okay.
Excellent. Pass it over to Bonnie for background.
Okay, so we can see on the slide, depressive disorders, it's debilitating, it's projected to be the leading cause of disability globally by 2030. And so just from the past, our systematic reviews as well, we saw that, for example, with ketamine and psilocybin, they are associated with alterations to EEG spectral signatures. And so here we kind of just thought, what if depressive symptom severity may mediate / moderate that association? So future research vistas can look into that, but here, as Anika said, we're just looking at differences in spectral signatures between high versus low severity depressive patients.
Okay, a little bit more about the data. It is pretty simple. It's data from the University of Arizona, specifically the John Allen Lab. And all they looked at is eyes open versus eyes closed EEG data from 122 college-age participants. As well, they looked at their Beck Depression Inventory Scale scores. So these participants scored reliably high, either high or low on the depression score. And I believe the high severity depressed individuals also had another depression rating scale done. So you can use that data as well to look at whether that affects the EEG spectral signatures or whether there's a correlation there. So our main question, again, reiterating again, is whether there are any EEG spectral signature differences between persons with high versus low depression severity, and only specifically in college age participants, which matters.
Okay, so some of the tools and methods are so the same, so GitHub and just Python and Biz and just the Toroto to actually look at some of the analyses, do some butterfly plots and just do some analyzing some plots and just functions of the data, as well as we'll be using R just to do some statistical analyses such as between group differences, looking at t-tests, seeing if high versus low have any differences in terms of just overall depression scores and things of that nature. So.
Okay, so since our data is only resting state EEG, we're just going to obtain the power of each wavelength, so alpha, beta, theta, gamma, delta for each participant, and Seb just said this, so between group comparisons on R or SVSS.
So these are just some of the deliverables. So then by the end, just to have the GitHub repository, the scripts, as well as just the graphs of the wavelength power, source organization, things of that nature, and just the write-up for all the statistical analyses through all.
There are references, mainly just the background that the tools that we'll be using. Cool. Questions?
So in the in the sleep deprivation, do you also have spectral power? Yeah, it might be interesting, like now, the more I think about it, I'm always interested in how much sleep and depression and mental health are related. So whether or not some of the signatures you see in depression are actually the same as you would see in the sleep deprivation study.
And probably in this population as well, because they're college students.
They're college students,
Yeah.
So
Yeah, there would be a.
high
At least it's like a homogeneous population,
Yeah.
right? So.
Even within the back depression inventory, don't you have a sleep question?
Yes.
Yeah. Yeah,
But from the analysis that we did, I think it's just like an MR image,
Yeah.
it's not specific. for
yeah,
each
okay.
Question,
Mhm.
so.
Even then you would need like a specific separate sleep questionnaire to do any
They're really,
sound analysis.
Yeah.
on sleep, yeah. So it doesn't really matter, even if you like separate
Mm-hmm.
the items on the back to questionnaire, it's not reliable.
yeah. Okay, but I'm not really- That's true. Okay.
I wanted to ask, so this is like college going participants whose data this is. What about like other variables that might have an effect on the outcome, like, you know, like substance abuse or like partying and like that have been reported in literature to have an effect?
So if anything varies, you go ahead then. No, it's just we have the phenotypic data set, right? So we get, if anything varies within that group of participants, we can control for in our statistical analysis.
Yeah.
Yeah.
And what test do you, or what method would you use to control for that?
Yeah.
Well, we're gonna use SPSS, we could potentially do, just GLM. Yeah, GLM, because there's no before and after, actually GE too. But you can set interaction terms on there and then you can add other, compounding variables.
Yeah, it might be cool for anything on S, you're gonna do an SPSS to try doing it in Jasper R to have. And then you can compare the results and make sure that you, yeah. That you have a like reproducible version.
Yeah.
Yeah, the same version on that.
Yeah.
Thanks, I'm asking these for my learning,
Yeah.
because I'm trying to figure out what to do with confounding.
What about analysis of covariance?
Yeah, you can do all of
Yeah.
that in SPSS. So we were
Yeah.
actually thinking about starting with SPSS because it
Yeah.
lets you just click on the covariance
Yeah.
and everything.
And GLM and GE also give you that analysis of covariance as you do it,
Yeah.
so, yeah, it
Yeah.
prints it all out for you.
And then as you mentioned, we can verify. it.
Yeah. Cool. Very cool. Yeah. Sarah, Jack. Sarah.
Okay, so, yeah, our group is gonna be looking at, we're gonna be, well, I mean, we previously heard about the MSC study, and so we kind of were interested about that, and then we decided to investigate and see how reproducible it is, and we're also very interested in longitudinal studies, so we thought that this would be perfect, and yeah, we're all undergrad students at the Kimmel Lab, and we're UT students. So, oh yeah, I'm Sarah. Oops.
Oh, I'm
I'm Jack.
Okay.
Hello.
So background, I think I mostly remember like Richie's mentioning this week, but like in terms of brain scans, like a lot of it is like based on like normative assumptions and like just the fact that like people think that a lot of our brains should look the same or like anatomically are the same. And then, but like in terms of brain scans, that's like really limiting because that doesn't really allow us to have like specific, like, it doesn't allow for specificity, detail, and then also clinical utility in regards to fMRI, resting state, functional connectivity. And so, that's where like this, the study that we're gonna be using comes into play because it'll allow us to investigate more the characterization of MRI, and then, in terms of like being more precise and that would be done by looking at MRI and fMRI data. And so specifically with this study, this data is collected from 10 individuals. And like it was collected by 5 hours of resting state with fMRI data, 6 hours of task fMRI, and then each scan was started at midnight, hence midnight scan club.
This is a nice visual summary of our background in the MSc dataset. So we have 4 different things: the structural MRI, resting state, and the task MRI, and the neuropsych batteries. Is very much like different like diagnostic tests like IQ score, cognitive tests, and personality tests. So each of the 10 individuals of our study data set were like underwent like this 4 different things. So structural MRI was like, I think it was 5 hours, and RSFT FC resting state is also 5 hours, and then 6 hours of task MRI.
So, when we were thinking about our main question, we were also thinking about the original, study's goals. So the original study had an emphasis on furthering the understanding of functional brain organization at the level of individual humans. So they were really looking at individual subjects rather than the whole group. And they wanted to see how consistent this individual brain connectivity was over time. So we wanted to see and test ourselves of whether we could accurately reproduce the results of this MSc study. Because of course, we also have to, within the lab, we'll also be doing our own longitudinal analyses, and we would need these skills later on in the future.
Yeah, so. In terms of data, like I said before, it was produced by the Midnight Scanning Club, and there were 10 subjects, and I believe those were like PhD students or graduate students, a mixture of them. And so there's 12 2-hour scanning sessions, and then yeah, there's like 2 sessions. It's divided up into different like sessions and different type of data that we can look at. But primarily like fMRI and also like neuropsychological testing. And we chose this because it has to do with fMRI longitudinal, and it's not pre-processed. Well, we'll be trying to like
We're dealing with
reproduce
it.
but yeah, so that's kind of.
The raw data.
Yeah, we'll be
These are the tools and methods we are planning to use. So we'll be making a GitHub repository with our coding scripts and like documenting everything. And the pipelines that we're planning to use are fMRI prep and MRI-QC. These are the main pipelines that were used in the original study as well. So, and also SIFT for the surface processing was also used in the main study. Also, depending on how long and like how big the data will be, we might have to use like competing clusters like Niagara. And yeah, and we'll also be making connectivity matrices.
So as we iterated before, our objectives would be learning how to run the pre-processing pipelines, working with Jupyter Notebook, GitHub, and practicing reproducible research and working on these longitudinal analyses.
In terms of deliverables, we would like to like redo the pre-processed data, and then also after processing, looking at connectivity matrices, and then like putting in using the skills that we've learned through BrainHack, and using GitHub repository and documenting our own code script.
This is the references for the first one is for the data set, and the second one is the original paper in the reference one.
That's about it.
You're open to critiques.
We're open to suggestions. We decided 1 hour ago. Might actually be, yeah, because you could see if we can get the individual people out of there. Or we should get the individual people out of the space. But it's just that there are only 10 people. Yeah, but they should be, like, the point is with that much data, that if you actually look at the like fingerprinting or the single subject connectomes, like in actual subject to subject space, they should be far apart.
Yeah. Yeah, that's
That's possible.
Or if you have,
Like, you should be able to know a person. Like, I guess we'll go back to like straight up fingerprinting, like you should be able to,
Yeah.
be able to find the person.
Yes.
The person's other day connectivity from themselves. Like, if you have one other connectivity matrices, you should be able to figure out
Doing the
which
reverse
one
of
of
the
the
stuff.
What?
Pretty much doing the reverse of the study.
Yeah.
Because like the studies like going
much
to
do
the
they change
functional
over
connectivity from the fixed, right? So you're gonna go from the functional connectivity to the other. Someone who
so that comparing, you mean
Comparing like how unique is the connect one for the individual people?
because everyone has like resting and separate tasks, right?
Mhm.
So I think it's on one of their paper where they show that,
Yeah,
I don't know which one, do they show that people cluster is better or task cluster is better? I don't,
and people cluster better.
yeah.
Yeah, if you can get through all of that, we'll see if we can get the. We hope we don't crash sign and get jobs that big.
Yeah.
That'll be the first step. And if we have to do tasks, like modify the scripts to do tasks at a time or something.
Or if
Yeah,
you're
right.
just taking connectivity, you could run the status too.
Oh, yeah, because you can run on the other side. So pre-processing lines, we'll see if we break sign up and we'll get that one for you. Or if it just hates it, rejects it. But then on the working with the connectivity matrices, there's a lot of questions that they started asking. So they started. Well, they started actually building individualized parcellations from this
Yeah.
data. But there's also like, if you have a parcellation, how many people with themselves, and how much do they change within themselves with different tasks, as different tasks happen.
That's the paper of Midnight Scan Club, mainly about when it first came out. So if you go to the paper, I think the title is something about individualized parcellation. So it's showing that people actually diverse a lot when you derive the parcellation
Yeah.
individually. And that's also what that group is pushing for, like emphasizing that the template might not be ideal.
Yeah. But they also have the stuff that they were the ones who had enough data to really say like,
Yeah, that's.
how much data do you need to have a unique picture of yourself? So it's like, do you need 1 hour data for it to be stable enough that it's gonna be, or can you use like 10 minutes from each day and still find yourself? Yeah.
Interesting question.
Yeah. Yeah.
Well, but it takes a lot of time to run an individualized parcellation of the whole brain.
Yeah, and I don't think we have this,
I don't think we have
Have
time to do that.
but you're gonna have the pre-process. So yeah, right, getting an individualized parcellation, I think you should probably use a so-called hard cookie cutter, like template parcellation. But we can go from a parcellated to connect, and then start playing with the connect. While we try to pre-process and not break sign it, or break sign it. Cool.
Okay. Let's move on to Marianne.
Yes, can you hear my voice?
Yes.
Uh,
Okay.
I'm
Should be able
sure.
to screen. And then the next one will be Shadi.
There's so many exciting projects. I'm so excited for you. Oh. Get a chocolate bar down.
Should I start?
Yeah.
So hi everyone, my name is Mariam and I'm a first year PhD student at U of T. And I'm happy to share the project that I'll be working on in the next 2 weeks. The title is Functional Connectivity Analysis using MRI and Neural Networks. So it's somehow similar to 2 projects that 2 other groups presented, but it's also different and I'm going to explain why. So a bit about the background, functional connectivity in the brain It is the measure of a bold signal in fMRI data, and it just shows that how different regions of the brain interact with each other. So with bold signal, we focus on the brain oxygenation level, and it shows that which parts of the brain are activated during the neural activities. So understanding functional connectivity just gives us insight into the brain function and also the neurological conditions. By use of machine learning, we can have advanced techniques to analyze and also visualize the complex interactions that are between the brain regions. So my main questions are going to be how can neural networks be used to accurately help us visualize functional connectivity in the brain using the fMRI data, and if we can create effective visual representations of the connectivity patterns that can help us better understand the brain function. So the data that I'll be using again is HCP or Human Connectome Project data. It has data on functional connectivity as well as a structural connectivity. I'm first going to focus on functional connectivity. I'm not sure if it's realistic to focus on a structural connectivity as well during these 2 weeks, but it would be good to somehow focus on both of them and see if the use of both functional and structural data will give us a better understanding of the brain function. So the HCP dataset, it provides high resolution fMRI data that is good for analyzing and visualizing functional connectivity. And everyone can access that very easily. So the tools and methods that I'm going to use, I will be coding in Python language with Jupyter notebooks. I'm going to use a range of libraries, for example, NumPy, Panda, and so on. And first, I will start by pre-processing the data to extract the time series and to make a functional connectivity matrix. Then I will use libraries for region-based time series extraction, and then I will calculate the connectivity matrices. And afterwards, I will use either CNN or GNN, which is convolutional neural networks or graph neural networks, to learn about the connectivity patterns. So my objectives are to develop a pipeline to pre-process the data and extract functional connectivity matrices. Then I will use them to train my neural network and analyze and visualize functional connectivity pattern. And then I will try to have a visual representations or a graph representation of this functional connectivity. And the deliverables are going to be documentation of the code and a GitHub repository. I will also use visualization tools in Python and present the results using them. And that's it. So if you have any questions, and I can't hear your voices clearly, so my apologies. I might ask you to just repeat your questions several times. It's not you, it's my ears. So sorry about that.
Any questions?
So with the Human Connectome Project, are you working from the minimally processed connectome? Like, are you working from the pre-processed outputs? Or are you trying to work?
I will try to process the data myself because I might not use all the features. I will actually use feature importance to first analyze like which features are the most important for my functional connectivity matrix, and I will only include those. So I will do the processing myself.
I think that's a step beyond. Because I think there's like 2 levels. Because there might be deciding your features, which is one step of pre-processing, but then there's just classic removal of noise features.
Oh, no, that's pre-processing. No, I won't do that one. No, I meant pre-processing for using like my machine learning algorithm.
Okay. So are you gonna work on the BrainHack copy or
I'm
accessing data?
I'm sorry, can you repeat the question again?
Locally, or are you planning to run, like, what system are you gonna run your machine learning model on?
What do you mean by what system?
Like, what computer?
Oh, okay, my own.
Okay. So you're gonna download the whole data set?
No, you can easily like have access to the data set using the Jupyter notebook because
Okay.
this open source data, you can just upload them on the working environment that you're using for your coding. Yeah, I don't need to
Okay.
download everything. Yeah.
Oh, and how are you bringing the noise confounds into your model? Wow.
Well, about the noise, I'm not sure what I'm gonna do. So there is the process of denoising of the data, but how it's gonna affect like my machine learning algorithm, I'm not sure about that. So I'm just gonna have to wait and see and then figure out something to.
Cool. Okay. All right, okay.
Thank you, Mariam.
Thank you.
Let's move on to Shadi.
Should I mute or should I be muted or?
Yes, you should be muted.
Okay.
Maybe we should move the owl so you're far away. I mean, should I mute the owl?
Yeah, we could do that.
Okay, maybe.
Is it? Is it fine? Is it fine now? Okay. Hi, I'm Shadi, and I'm at the end of my first year of my master's at York University, a Kohler Visual Neuroscience Lab. Actually, what I'm going to work on for my thesis is a behavioral one that is completely different from here, but I try to be prepared for my future actually studies. And in terms of this project, I'm going to focus on SPN. And in terms of background, I should say that visual symmetry processing is a fundamental aspect of human perception involving automatic detection mechanism. And the brain's response to symmetry can be measured using ERPs, such as the SPN, which is sustained posterior negativity. And as you can see at the bottom is some of the pictures of this article that I'm going to simulate for this project. And nowadays it is a hot topic to focus on special attention beside visual symmetry and how it affects this SPN. So it is said that special attention can modulate the SPN response to visual symmetry even in an unattended region of the visual field. And so understanding how the brain processes symmetry and attention provides insight into the mechanisms underlying visual perception. Actually, my main question is, are contralateral SPN amplitude generated by symmetry at attended and unattended spatial locations different? And in terms of data, in this, actually, I got my data from the article that you can see at the bottom of the screen. And there were 48 participants, and there is an EEG data, and which is recorded by using biosemi system, and also there is there's data collected by infrared eye tracker, and on bottom right, you can see actually the stimulus. There will be random and also symmetric patterns that people pay attention to them. And I haven't gone through all of the details, but this is the whole idea. And they were actually calculate the amplitude of SPN in different situations. And as I told you, it is considered contralateral. And the method that I'm going to do is actually the referencing and the EEG over the skull and the low pass filtering and segmentation around into epochs. And I will use ICA to clean the epoch data and remove artifacts and isolating the neural response that relating to visual symmetry processing. Actually, by using this method, I wanna learn how to use modules learned here at BrainHack to analyze EEG data in visual neuroscience of symmetry. These modules are Python coding using Visual Studio, EEG data analysis methods, and visual data visualization and statistic analysis using R. And what I will be handing is a documentation describing my applied analysis pipeline and also my Python code script. And this is the reference that I'm going through and work on it. Actually, I'm using on VS. Yeah.
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 Wait,
Yes.
Yeah. And you already have the data?
Yes, I used the data from the article that I mentioned. Sorry, I cannot. Okay. Okay, stop sharing. Okay, sure.
So we have Aisha next, and then followed by Flavia, and then Alice and Yassine.
Hi, everyone. My name is Aisha. I am a graduate student at the University of Toronto. Just a disclaimer, my research is actually on the spinal cord and not on the brain. So this is a new area for me. My personal objective for joining the BrainHack school is I want to learn new skills and practice them, and also I do really like the brain. So will this story on its own? No, it's just right on my screen. So we do know from previous studies that age has shown association with the anatomy of the brain, where usually we notice age-related atrophy. As for gender, the definition of gender itself has undergone changes over time, because previously sex and gender were considered the same thing, and so they were documented interchangeably in older studies and clinically as well. But now we try our best to document them separately. And they have shown some association with anatomical differences, but those studies tend to often be controversial. Additionally, these are also two variables that are most commonly documented clinically and in research. So it would be interesting to see if they have any association with the functional anatomy of the brain. So my main question is that does age and or gender of an individual affect the functional connectivity in the brain? And for this, I'm going to use the Midnight Scan Club. So I'm very grateful for the 3 individuals who already described the data set. It's just like them, I probably made this like slideshow like 20 minutes before the presentation. So it's great that they did it because I actually did not get the time to describe it.
They're all the same, same data set.
Yeah. And from a brief glance, they have age documented, they have gender documented, and they have fMRI documented in detail. This is open data available using brainlife.io. And hopefully I will use these tools. I'm not sure why I'm using GitHub other than practice because it's just me on the team. But I am very excited to learn and practice the skills through the modules and apply them to this data set. So I have not decided which statistical tests I would run, but most likely t-test for age and chi-square for gender and association with functional connectivity, which I would start with resting state functional connectivity. And then once I get the hang of it in the first time, I would look at task-based connectivity. These are the very hopeful deliverables. So documentation, most likely GitHub repository, maybe markdown file, most likely, and maybe a code script as well. And the rest is the reference for the data set. That's all I have.
For the GitHub, if you want, because this is an open data set, right? So you could just work on the BrainHack school organization and put your project there. And at that point, you need to collaborate with others to get it published on the website.
Anyone who ever gets free time and wants to help me learn with the skills, please do so.
Anything else? No. I do wonder if you do get it. It might be interesting to see if you could transfer your scripts to another data set quickly. Because like age and sex assigned at birth should be something that we see a lot across a lot with the data set that exists on, I might be able to get similar brain metrics too if it's preprocessed and just see if you pull out the same, if you can replicate actually across data sets once you have the script built. That would be interesting. Yeah.
Okay, nothing. Okay. Gloria, you're next. Maybe unmute yourself. So I could put you.
All right, so hi everyone, I'm Gloria. I'm also at the University of Toronto, but I'm faculty of music, strangely. But I do music and health science / neuroscience research. So I'm based at Li Ka-shing Knowledge Institute, which is also the research center for St. Michael's Hospital. So yeah. So most of us in this room right now, especially, are probably experiencing this. So before caffeine, we probably feel a bit like this. And then after caffeine hits, we might feel like this, a little bit happier. So aside from the obvious physiological withdrawal symptoms that we often experience, the most obvious thing that we notice when we don't have enough caffeine in our system, let's say, is that we feel exhaustion, which leads to a reduction in motivation, therefore poor performance and learning outcomes later on in the day. And so in this study, I will be, well, this study is actually inspired by an fMRI paradigm that was published by you and colleagues back in 2012, but this is specifically a EEG plus FNAS one, which I'll break down a little bit later. So it's natural that how we feel in this moment can largely impact our ability to pay attention, to learn new skills, to learn new information, but most importantly, how we memorize or even retain those pieces of information, as well as other higher cognitive processes or cognitive skills. And so the effectiveness and efficiency of our learning can vary greatly depending on kind of which end of the spectrum we're sitting on. And this kind of motivational intensity can also be referred to as the broadening or narrowing of our cognition. And so all of our reward-driven behaviors, such as your learning, valuation of value, I suppose, and decision-making, these can be divided into 3 sub-processes. So firstly, generating that motivation, secondly, maintaining it, and then regulating that motivation. And so let's have just a quick little boxing match between EEG and FNAS, because I know not everyone's very familiar with this particular modality, but let's start off with fMRI since most of us are familiar with this. So fMRI can look at smaller or more deeper structures in the brain, and it overall has a better spatial resolution than FNAS or EEG. But the disadvantages of fMRI come at the cost that it has has low contrast to noise ratio of your bowl to your blood oxygen level dependent signals, and you can often have image distortion, signal dropouts during the scans, and obviously it's very expensive. Whereas for FNAS, it is low cost, it's silent, literally, it's transportable, so it's very mobile. So, you know, that makes it quite convenient if you want to monitor long-term changes or do repeated measures of certain cortical activities over a period of time. And so it makes a great alternative to fMRI because you can still investigate these human cortical functions. But the disadvantages of FNAS is that it has a low temporal resolution, but it's still better than fMRI. Yeah, but it has local hemodynamics only, so you can only look at local networks, and overall just has shallow penetration of the skull because it uses infrared light. And then if I compare this over to EEG, so keep in mind what we know about fMRI already. EEG has more tight temporal resolution, so you can take about 500 to 1,000 samples per second. And EEG measures the electrical signals originated directly from these active neurons. And the near system measures also the change of oxygenation resulting from metabolism of the active neuron. So it's really at the intersection of EEG and fMRI. Well, FNAS is at the intersection of EEG and fMRI, sorry. But EEG also has its disadvantages. It also has a low spatial resolution because the electrodes can only measure electrical activity at the surface of the brain. So, yeah. But with FNAS, like I said, sitting at this intersection between fMRI and EEG, it shares various commonalities and differences. This is why this multimodal imaging method of FNAS plus EEG is essentially a match made in heaven. So often we can actually now do parallel acquisition, doing both at the same time. So you have this kind of swimming cap looking thing that has both the EEG electrodes and also the FNAS on top. So this particular experiment, not my data, by the way, full disclosure. So the experiment was divided into 2 parts. So the cognitive motivation task and also the recognition task. So this is kind of their basic demographics. I won't go through that. It's pretty self-explanatory. The participants basically can freely choose whether they want to remember a specific scenic stimulus. So they're going to get shown about 500 photographs, and then after they see it for the 3 seconds, they can decide, do I want to remember it? Do I not want to remember it? So that's their self-motivated behavior. And then the results from that recognition test were used to determine whether those participants could actually recognize that scene later on. aka measuring their memory or their learning ability over time. And so from the experiment, the trials from each participant can be divided into 4 categories, or at least that's how the authors decided on it. And I'll break this down on the next page for you, but just a quick look of what this experiment actually looks like. I'm sorry, it's so unclear. The picture is really unclear. Try to find the highest resolution one. No. So basically, this is the attention period. You got 3 seconds where you look at the picture, and then this is your decision period of 9 seconds where you can decide, do I want to remember it or do I not want to remember it? Up to you. Full autonomy goes to the participant. And then once again, shown another picture, and then they have another decision period, so on, like that. Okay, so basically it's broken down into 4 categories. So want to remember, and then they manage to remember later on. They want to remember, but they ultimately forgot, and vice versa. So hopefully that kind of makes sense. Although it is honestly a bit of a confusing protocol, I'm not gonna lie. So my main question is obviously based around the data set itself, because it's not mine. But I think from this, we can look at how motivational states impact the recognition and memory performance, because we have to be careful when we talk about performance and learning outcomes as well, because performance is without a retention period, like without delay. Whereas if you're looking at learning, you have to have that period of no learning and no interaction with the stimulus at all. So usually that's about 24 hours in most studies. Okay, so like I said, I'm going to use EGFNER, so both at the same time. Very ambitious, I know. I'm going to try my best. But why? Ultimately, I want to focus on the cognitive responses, event-related potentials to the stimuli, and also just learning how to look at this combination of modalities together. And this data set was taken from Mendeley data, because that's really one of maybe 3 data sets with both EG and NES, so kind of had no choice on this one. And like I said, it kind of shaped the research question around the data set itself, because not my choice. Yeah. And with the data set, it has both raw data and pre-processed, but I like to make my own life hard, so I'm going to do the raw data and go through the entire process. So, okay, in terms of tools, well, most of the tools available now are through MNE Python, and they're compatible with most NES devices, most asterisks. Alternatively, there are also various toolboxes through like MATLAB, so your Homer 2, Homer 3 packages, and also NES SPM. Homer, not as in Simpsons Homer, but yeah. And of course, for non-computational tools, I actually will be humbling borrow Mina Rose's genius brain to figure out how to actually hack through this whole FNES thing. So yeah, it's gonna be a collaborative effort of some sorts. These are kind of the objectives. I can always finesse them and always reduce them, but basically looking at the data set itself, we can understand how functional activation patterns and network activity in the brain may differ between the different self-motivated behavior. So deciding to remember or deciding not to remember, and also to investigate potential mechanisms underlying those brain states. So feeling good or feeling bad about wanting to remember or not. And also last, examining how the potential impact of self-decision-making processes may enhance or suppress the resulting recognition, so remembering that picture during that cognitive motivation task. And once again, not study related, but personal goal is to learn how to kind of map one onto the other. So yeah, and depending on if I go down the route of using R or Python, I'll hopefully have some sort of markdown file or code script for future analyses, because my lab also has no pipeline at this time, because most systems don't come with a pipeline, so you have to write your own. So yeah, I guess that's on me. That's the references I used, and yep, thanks for listening.
I didn't know that MNE had it years.
Yeah, so they have the EG toolbox, and then they also have,
They have MNE.
yeah.
Which is good. At least MNE.
Yeah, well, yeah.
Question about the task. Do they have anything to say they just
I think it's keys on a little, yeah.
Do you filter through like, cause what if they just decide to remember?
I don't think they did. I had a brief look and I don't think they did. But my problem with this is that obviously there will be uneven numbers in terms of.
They might be not have
Yeah, I mean, it's 500 pictures, so. I know, it's not my study. If it was up to me, I wouldn't do that, but.
That I would assume would be important are not near the outside of the brain.
Yeah.
So not gonna be near the cap.
I guess that's why you have both EEG plus apnea.
Both EEG and apneas captures better like towards the scope.
In general, yeah. Although apnea is kind of between EEG and fMRI, so it has still better spatial resolution than your EEG. So it'll pick up more information, but yeah.
Cause like your ventral visual stream is still gonna be text clear.
Basically.
In the middle of your head, in your hippocampus.
Still very surface, yeah.
They are right in the middle.
Yeah, still very surface, but
It's potential.
yeah, I mean, imagine if we can capture the parahippocampal region well, that would be nice, but yeah.
Okay, Alice, thank you for staying this late.
It's all right. Okay.
Have you?
I guess I will screen share.
Put the speaker back on.
Say something? Can you say something?
I think your mic is open.
It should.
Can you see the slides?
Yeah, we hear you. Okay.
Okay.
Do slideshow. The slideshow.
Okay. All right. So Yasin and I are planning to do white matter development comparisons between phenotype and non-phenotype children and teens. And phenotype means there are few well, two specifically ADHD and DMM. And sorry, DMDD. So these are mood disorders, and they are all related to irritability, attention, hyperactive of the children. And we want to see how does the white matter changes over time. So there's some, we're using the same data set that has been presented in the reference number 1, which is saying there are difference in the gray matter volume, but there's no indication on the white matter. So we want to see on the structural connectivity side, is there any differences between the healthy control for as time progress. So again, the main objective is only looking into the differences between phenotype and non-phenotype structural connectivity progress over time. And we use age as the time. The main question here is, does the white matter vary? We know the gray matter varies comparing to the phenotype, meaning the ADHD and DMDD kids. So now we want to see on the structural connectivity side, does it also vary? And we use the white matter trajectory to determine. And the data set we're using is from paper 1, which is published in Open Neuro, the number reference 2. And here, they actually have 521 participants, but only 152 has the DTI samples. And the age group is from 9 to 20. And if you look at the breakdown here, it is a little bit white dominated. Is 73% of the scans are from white. Comparing to non-phenotype and phenotype, there's only 40% which are healthy control. I think it's still okay. It's not too much of a difference. And the tools we are planning to use, so what we are planning to do is group them by age and then compare the two groups, phenotype and non-phenotype. And we are using the DWI scans from the data set. We're going to use QSI PRED for pre-processing and reconstruction as well. But we're not familiar with the reconstruction process. So we will also use, hopefully we have time to use this TriTrack. TriTrack is a tractography tool as well, and to see the differences in the results. And the overall result is to compare group averaging of each age between the two groups, and to see how the white matter progress, how the white matter grow over time. And it's a very simple project, so we do some illustration and write up what we did and how we do it, and the analysis in probably in a bioarchive format, and just put it on bioarchive. And these are the 4 simple references, the 2 papers and the two tools. And that's it. Any questions?
Oh, I have a question about your age. Are you grouping the participant based on their age, like each year?
Yes, based on the age, because there's only 1 scan per person. So we cannot really track the growth of individuals and try to averaging the growth, but we just do stratify by age, and within each group, I mean the phenotype and non-phenotype group, we have a subgroup by age.
Will your age be, let's say 9 years old, 10 years old, 11 years old, and so on, or?
Yeah, exactly. Yeah.
I'm thinking maybe it's worth binning them. So
Yeah.
and also there might be some like delayed or, individual differences in the development, which might not be that meaningful. Let's say if someone is just slower and developing at year 10, it doesn't necessarily mean that there's a difference between year 9 and 10. So maybe that in some way will help, and that will increase the sample of each group as well.
Right. Yeah, the thing is this is a very unusual data set, because if you look at the IQ, right, even though the age group is 9 to 20 years, but the mean IQ is 111, even though the standard deviation is 13.2. This group has way above average IQ.
Yeah. But you also have people going from elementary school to like undergrad.
Yeah, that's true. Yeah. Or maybe originally we were thinking only look at the teenage age, because that is kind of the more obvious spruce, the growing spruce. So we might see more significant differences there. That also can sort of but the thing is, if you have more granular, like temporal samples, you get finer temporal resolution, you get better better plotting over time. So it's a trade-off of sparsity of the data points where we have. 152 is really not a lot. Like you said, divided into 9 to 20 years old, and this is over 12. We get probably 5 per group for each age. And it is very, very few. That's true. The thing is, we haven't really looked in depth yet in terms of how the distribution of the participants age yet. So it's really hard to tell. We might be in, let's say, primary school, as one said, it really depends on what we get in the scans.
I think maybe instead of grouping based on having equal sample size for developmental data, it might make sense to group them based on like the actual time of development, right? Like before teenage years, during teenage years, or after.
Okay.
Did they also put out puberty,
Yeah.
a puberty variable or not?
I don't think so.
Not that I love self-report puberty, because it is a terrible scale and little boys just look at it and laugh. They're like, I see me, I have hair there. But it surprisingly can be sometimes more related to some of these brain variables than.
Right.
So.
The hormone effect, right? Yeah. Yeah.
On the brain development.
So you
Yeah.
can estimate that with like roughly with years.
Yeah, like it's gonna be correlated with age too.
Yeah.
Well, we do have sex.
Oh, would you?
Yeah, you will
You
will
group
see
them
sex.
differently?
You will see sex differences too, but it'll be hard because in your phenotypic data is probably not balanced. Like ADHD, you probably have a lot of boys.
Yeah, it's 60%, almost 59.2% male in the scans.
Yeah.
Yeah.
It also doesn't surprise me that the average IQ is high, because that's just. It tends to be like convenience samples, like if you start asking the moms of the scientists to bring the kids in, you start to get like the more privileged, like 110 IQ average population.
Like the sample said, like people who make money more than 90,000 are actually more.
Yeah, look at the income, right?
Yeah.
If 40% is less than 90, and you're like 60% over.
Wow. That looks like a lot like scientists income.
It's
like not high.
Yeah, but it's like the scientists decided to advertise the study out of their kids' private school.
Like blue collar workers might not want to bring in their kids, like.
Yeah.
Because they might also be a bit hesitant that their kids are sometimes already facing barriers.
Yeah.
If they have a documented, condition, then they do worry that they might face even more barriers, like getting into school and stuff. So that's the social barrier. I had a question about the college going thing. So between 18 to 20, or even like 17 to 20, a lot of people would be transitioning to undergrad or going to undergrad, and with ADHD, just in that context, that would mean that they're exposed all of a sudden to very new stimuli. Would that like affect things, like confound things with like compared to someone who's like 10 or 11 with relatively stable like school and that environment?
Yeah, that's why originally we were thinking only using teen, like 14 to 16, that age, the growing spruce age, and trying to see the differences. But the data set has that whole range, then why don't we just take a look first and decide later?
Yeah.
And like I said, it's really white dominated data set. It's 73% white. So it's a bit biased data set.
Just out of curiosity, have you take a look at the actual data yet? Like all the details in the data?
Yeah. It's really one scan per person.
Okay. And do they have more variables?
Yeah. Yeah, it has the phenotypes. So besides the participants TSV, this is basically just male, female, and it's not down over here. So it has this participants, age and sex, but there's also the actual phenotypes. So this one has more information. If you look at the individual session, it only has one scan, one session, that's it. And the phenotype itself has a lot more information. Like you have the actual phenotype labels. But like I said, it's just phenotype. It has nothing to do like with like puberty or anything like that.
I think, you have you have KSAD. This is
Yeah.
also.
It's different batteries, yeah.
They have CPRS, is that a scale? A questionnaire for?
Actually, if you go back to, so this is if you go back to the phenotype JSON.
This is okay. This is the phenotype TSV is the same as JSON.
If you go to the JSON, and actually you can hit the view to, I think the view should work. Yeah. Oh, okay, that's the participants JSON.
Oh, sorry. Phenotype. Oh, where's the phenotype?
Yeah. Oh, okay. Yeah, so this one, if you actually click plus, because it's actually giving you kind of JSON viewer, this is the magic of bids.
Oh, that's nice. I didn't know that. You can just
if plus on it, then it will tell you information. It'll give you more information about what each column is.
oh.
Yeah, I think if you click on it, yeah.
Oh.
Oh, see, look at the
And
question
levels.
something, yeah. And levels will show you how it's coded.
Oh, I didn't know you can do that. Oh, this is nice. Because I look at the JSON file, it looks like just like the CSV, except
Yeah, but yeah, you have the company JSON, so you can start to understand what's in the CSV.
okay,
So
okay.
uploading, but still helpful. So helpful
This is like even this, I think this data set, if you don't do neural imaging, you can do it just do metadata analysis, right?
Yeah.
Just do the actual structure data analysis and that's it.
Are you planning to use any of these questionnaire data?
Don't know yet. The main focus is still try to learn how to analyze DTI for me. But I think this data set is so rich. The paper itself already, they use the bilinear model, like similar to the one you presented yesterday. And the paper itself has the outcome. So it's interesting. Where is the paper? Sorry, 1 second.
Yeah, because
I have it somewhere.
once you have the DTI,
Mhm.
it might be interesting to look at how they relate to. If you could get some summary scores from the questionnaires,
Yeah.
then
Yeah.
that will tell you more than age or other things.
Age is still a super strong predictor.
Yeah.
So I would imagine it would still be a strong contributor.
So the paper itself has some analysis already. It's just I haven't really gone through it in detail. Let me see.
Have any pretty pictures?
Oh, yeah, lots of pretty pictures. So it's the effector loadings for irritation and attention hyperactivity. And I don't know what this is doing here, but look at the pretty pictures. So latent factors with independent syndromes. So it's exactly like what you were doing yesterday, right?
Yeah.
And they do have like different scales, and then so Y, right? And then I don't know the latent factor. What is that? I have no idea. Oh, share factors. Okay.
Okay. Or DWI data, or is this just on the phenotypic file?
I think this is only phenotypic. I don't think they did the DWI. Maybe later. 1 second. See, that's the picture I asked you. Remember last week? Last week, was it last week? Okay. Isn't that pretty like circle, right? How do you plot circle? Okay, yeah, they do use kind of a
Oh, so yeah, they did it against the TBSS.
yeah.
That's cool. Yeah, you could try to replicate. Actually, yeah, I'm still trying to figure out how to do back factor.
It's a very, very interesting paper and data set to read. Like, yeah.
How
But for-
do they deal?
Pardon?
Oh, in this paper, do they incorporate age?
Yeah. Do they just? Yes.
Good question. I don't think.
Oh, wait, they have an age down there. Where'd it go?
If you scroll down the scatter plots, the
Age, mean effect. Oh,
Okay.
age, definitely, that's surprisingly strong, like punch you in the face relationship between age and like fractional velocity, which is coming from the DPI database. It's true,
Right.
DTI punch you in the face with their age relationships.
Yeah.
Like, oh, I guess, okay, we can go home now.
Okay, they did the analysis. Okay, like I said, it's just a practice, right? To do DTI.
So maybe you don't need to group them according to age. You could just use it as a continuous scale.
Pardon?
Right? You could just use it as continuous numbers.
Okay, like a covariate.
Oh,
Yeah.
they're doing like better grading. Like, oh, sorry, yeah. Okay, yeah, because they're taking the residuals of a like age linear model and running it against their behavioral factor.
Mm. Mm.
Do it anyway. Okay. Oh,
But it still doesn't answer the white matter.
yeah.
I think this is on gray matter. I don't think they use DTI, do they?
No, that's white matter.
FA is white matter.
That's white matter.
Okay. All right, I didn't see that. Okay. I haven't finished reading it.
Like masking the deep white matter instead of doing tracks. Yeah, it would be interesting to do tracks.
Yeah.
Give you, or you get things different across different areas of the white matter.
Right. This is kind of a funny spot of white matter. What is this like anatomically?
That's a good question.
It's the singular place.
Singular jar.
Is that singular jar?
Like on this section, it looks like that.
I think it is. Oh, but it could be.
It's near.
It's hard to tell, like, oh, that's the water script.
Looks like a corpus callosum.
Yeah, no, corpus callosum.
No, that's the
there.
corpus
So it has-
Oh, on the medium one, yeah, that's true.
But this is probably stuff that's going on here, but probably our.
It's such a
really
Yeah.
Oh, okay. I couldn't hear what it is, but
Okay.
yeah.
Okay. Running away. Okay, we're in the Mars building on Monday. Don't come back here.